Hello everyone, myself Zeel Bhatt and welcome to my channel Zeel's Pharmacy Tutorial. And today we are going to study about DNA replication. So first of all, let us see what is DNA replication. DNA replication is a process in which DNA copies itself to produce identical daughter molecules of DNA. So now let us see what are the steps involved in the DNA replication. There are total 7 steps involved in the DNA replication. First step is formation of untwisted DNA, formation of replication fork, formation of RNA primer, then fourth is addition of DNA bases, then removal of RNA primer, addition of DNA bases, and the last and the 7th step is twisting of DNA into helix shape. So the first step is formation of untwisted DNA. The helix structure of DNA is untwisted and hereby the process of DNA replication is started. Now the second step is formation of replication fork. The untwisted DNA comes in the contact with the enzyme DNA helicase, which is a zip opener. The untwisted DNA is divided into 2 templates and thus results into the formation of replication fork. Now the third step is formation of RNA primer. The enzyme primase is responsible for the formation of small fragment of RNA which is known as RNA primer. The 2 templates formed from the replication fork can be classified as 2. First is the leading strand and the second is lagging strand. The leading strand runs from 5' end to 3' end and the lagging strand runs from 3' end to 5' end. The RNA primer formed from the enzyme primase binds to the leading strand. It also binds to the lagging strand, but a continuous supply of RNA primer is needed for the formation of DNA in the lagging strand, whereas only one molecule of RNA primer is required for the DNA replication in the leading strand. This point is considered as the starting point of DNA replication. Now the fourth step is addition of DNA bases. The enzyme DNA polymerase adds the DNA bases in the direction of 5' end to 3' end in both leading strand as well as in the lagging strand. There is continuous supply of DNA bases in the leading strand, but on the contrary in the lagging strand, which runs in the direction of 3' end to 5' end, the DNA bases are added in the direction of 5' end to 3' end. This results into the formation of small fragments which are known as Okazaki fragments. Now the fifth step is removal of RNA primer. RNA primer which are attached to the both leading and lagging strand are removed in this step. This is done with the help of the enzyme exonuclease. Now the sixth step is addition of DNA bases. DNA bases are added in the place of the RNA primers. This is done with the help of the enzyme DNA polymerase. This results in the formation of untwisted daughter DNA. Now the last step and the final step of the DNA replication process is the twisting of DNA into helix shape. This is done with the help of the enzyme DNA ligase. Untwisted daughter DNA molecule is finally twisted. Now there is one important thing about DNA replication, and it is that the DNA replication is semi-conservative. This happens because the 2 strands of parent DNA is conserved in the daughter DNA. This could be easily figured out from this diagram. So, DNA replication is a semi-conservative process. Thank you.